Hey, Amy here, back with part 4 of section 7.1. We're going to finish up this section, and we want to look at how DNA makes a copy of itself, and also about the 3 different types of RNA and how RNA is different than DNA. Okay, like in the previous video, one of the things we mentioned about DNA is that it needs to be able to make a perfect copy of itself, because every time your cells are dividing and splitting into 2, they need to pass on a copy of their genetic information. So DNA needs to be copied quickly and accurately. Okay, the process by which DNA replicates itself or makes a copy of itself is called replication, making a replica, exact copy of itself. Now, in order to build it, there are many free nucleotides kind of floating around in the nucleus. This comes from the food we eat. Okay, we break down the DNA in the food we eat, the cells we eat. So we've got, A, T, G, and C, and phosphate groups and deoxyribose sugars. Okay, and those nucleotides are floating around so that they can be used to build new DNA molecules. So the steps. Step 1. The DNA helix untwists. Okay, there's a special enzyme that takes this DNA, and you'll see it in the next picture on the next slide, and it kind of untwists the DNA and actually has to kind of cut it and let it spin a little bit. Otherwise, if you've ever tried to untwist a garden hose or something like that, you'll see that it kinks. And so there's an enzyme that helps this untwist. And then there's a second enzyme called helicase. Helicase, you can see it right here, unzips the DNA. By unzips, we mean it's going to break the hydrogen bonds down the middle of the molecule between the nitrogenous base pairs. Okay, so it's going to break those bonds and peel them apart and expose each side of the DNA. Okay, step 3 is that we need to build the new DNA strands using the old strands as a template. Okay, the enzyme that does that, another enzyme now, is DNA polymerase. Okay, so practice saying that, DNA polymerase. Okay, polymerase, now there's a trick. It can only build, synthesize means to build in the 5' to 3' direction. Okay, we're going to talk a little bit in class what this 5' and 3' means when we talk about the DNA molecule. But you remember that it's anti-parallel, so they actually run in opposite directions. And DNA, or the DNA polymerase, is only going to build in one direction. Well, because they run in 2 separate directions, when you split this DNA apart, so here's the DNA helicase. Okay, the old strand of DNA here is 3' to 5'. Okay, the new strand can be built 5', and here you see it twisting around and building this way. Here's DNA polymerase, the enzyme, and it's going to build in this direction. Okay, it can build straight along. So as the DNA is unzipped, this DNA polymerase is going to go along, and it's going to match up complementary bases. Okay, so if there's a T on the original strand, it'll put an A. If there's a G, it puts a C, a C, a G, and so forth, and it just reads along as it unzips all the way till it gets to the end of the DNA strand. On the other side, we have a special case, however, okay, because now it's running, it has to build backwards. It has to build in this direction. So what happens is, as it unzips, we always have to put a little bit of RNA primer down, okay, in order for DNA polymerase to start building, and it will build in the reverse direction here. And as it unzips, it jumps in and builds back, jumps in further, keeps building back. And so it keeps having to build back. And so you get these little short pieces here, like here, okay, and here. Okay, these little pieces are called Okazaki fragments, named after the Japanese scientist that figured this out. And so all these little pieces, there's another enzyme, DNA ligase, that comes along and kind of seals these together, takes the RNA primer out. Let me get rid of some of my marks here. Takes the RNA primer out and will seal them together. So here you see the DNA ligase putting all these little Okazaki together. Now, obviously, that takes longer than just reading straight down through there. So the one that reads straight through we call the leading strand, because that can go much faster. On the other side, where it has to jump in and read backwards, we call the lagging strand, because it takes a little bit longer for that to happen, and there's more pieces and parts kind of jumping in, building back, sealing off pieces, jumping in again, and so that's the lagging strand. Okay, make sure you kind of label this in your notes and star some things because of the things that I circle here, you may see again. Like on a test. Okay, so make sure that you know these pieces and parts of this DNA replication model. Okay, step 4. Now that we have the new base pairs built using the old code, bonds formed between the nucleotides. We call them phosphodiester bonds between the phosphates of one nucleotide and the deoxyribose sugar of the next nucleotide, and they all get strung together so that when you get done, the new molecules twist into the double helix. We have what we call the semi-conservative nature of DNA. In other words, you conserve each strand. So originally in blue up here, the original of replication, notice the new strand now is in pink. So you have the first generation daughter strands have half blue, half pink. When we go through round 2, okay, round 2 of replication, when those each divide, now look, now those are going to split. Now you only have one blue out here, one blue out here, pink, 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 pink. Now you have brand new DNA down here. So every time this divides, it's saving the old strand as a template to build the new strand. Okay, that helps ensure that you're getting an exact copy of that original DNA strand. Okay, some final thoughts from the first section here is this idea of RNA. Okay, make sure that you understand that RNA is ribonucleic acid. Okay, it's where we get our RNA from. Okay, RNA helps get the message from the nucleus out to the ribosome and then also helps build the proteins. There are 3 differences that how it is different from DNA. The first one, it's single-stranded. Okay, here you see DNA, and it's that double-twisted, double helix strand. Okay, RNA, just a single strand. Okay, sugar to phosphate backbone with the base pairs kind of sticking off, exposed in the middle. Number 2, it contains the nitrogenous base uracil instead of thymine. So over here on DNA, you see it has thymine, which is a pyrimidine. And then over here, it's replaced in RNA by uracil. Okay, and this will be important when we get into the next section. We'll start using U or uracil instead of thymine, and you'll see how that works. And the third difference, it contains the sugar ribose instead of deoxyribose, hence the name, ribonucleic acid instead of deoxyribonucleic acid. So you can see here the backbone here that has the phosphate sugar backbone. The DNA is a little bit darker colored because that's deoxyribose, and they made this a lighter color, okay, to represent that it's a different sugar in that backbone. And finally, there are 3 types of RNA that we're going to discuss in this chapter. The first type, messenger RNA, also written shorthand as mRNA, okay, is what helps take the genetic information from the DNA through the process of transcription out to the ribosome, okay, where it's going to be made into the protein. Okay, so DNA to mRNA is known as transcription. Okay, so that, remember, DNA is locked away in the nucleus, and so it has to be transcribed into a message which can be taken out to the ribosome. The ribosome itself is made up of RNA. Okay, down here is a picture. We often abbreviate it lowercase r RNA. This is our mRNA, single-stranded. rRNA is a ribosome that's made up of RNA, and it also has some proteins, and it has a large subunit and a small subunit. And it's going to be used to read the mRNA out in the cytoplasm and assemble the protein. The third type of RNA that helps actually build the protein is the tRNA, which we usually use a lowercase t for transfer RNA. These things right here on the end, this little ball here on the end is an AA amino acid. They go out, they have an anticodon, and we'll get into this a little bit more in the next video. But they have an anticodon, okay, which matches up with the codon or the code here on the mRNA, and they read the code on the mRNA, which originally came from the DNA, and they're brought in here to the ribosome, and it just kind of all slides along, and the tRNAs bring in the amino acids and join them together and build that protein. Because remember, proteins are built from amino acids, and they're read with the tRNA, the rRNA, and the mRNA. In the next part of the chapter, we'll look at how all these three types of RNA work together to get the code from DNA and build and express that gene as a protein.